Hello everyone, welcome to my channel, and today I'm going to talk about part 2 of the cell topic. In case you have missed the part 1, I will link it in the description below. Please check it out, and if you have any doubts you can always leave your questions in the comments below and I'll get back to you. And in today's class I'm going to talk about ribosomes as well as cell membrane. And I'm going to talk about cell membrane and the fluid mosaic model. I have also made a short video on the fluid mosaic model before. In case you have missed it, please check that also. So let's get started. So in the previous class, I have given you an overview of the cell as well as a description about prokaryotic cell. So today we'll be talking about ribosomes and eukaryotic cells and cell membrane. All right. So coming to ribosomes. In prokaryotes, ribosomes are associated with plasma membrane of the cell. All right. So how does this ribosomes occur in prokaryotes and eukaryotes? So in prokaryotes, they are associated with plasma membrane of the cell. All right. And what is their size? Their size is 15 nanometers to 20 nanometers in size, and they are made up of two subunits, that is 50S and 30S, and together they form 70S ribosomes. Okay, so they are mainly for protein synthesis. All right. So don't get worried about your NCERT textbook. I'm actually explaining from the NCERT textbook. So these are like condensed notes from the NCERT textbook. So you can follow from page 129 from your NCERT 11th class textbook. And in case you do not have the textbook, you can just listen to this presentation and I will leave the PDF link to the notes below. All right, in the description box at the end of the lecture. So let's get back to the topic again. We were talking about ribosomes, that in prokaryotes, they are associated with the plasma membrane of the cell, and they are 15 nanometers to 20 nanometers in size, and they are of two subunits, 50S and 30S. Together they form 70S, and they are the site for protein synthesis. And let me tell you something interesting. Several ribosomes attach. So several ribosomes attach and they form a single mRNA. And this chain of several ribosomes attached together is called as polyribosomes or polysome. All right. And the ribosomes of this polysome, what do? Is they translate this mRNA to proteins. And this process is called as translation. All right. I repeat, there are several ribosomes that are attached to the single mRNA and they form a chain that is called as polyribosomes or polysomes. And the ribosomes of the polysomes, they translate mRNA into proteins and this process is called as translation. Now coming to inclusion bodies. All right. So what are these inclusion bodies? Inclusion bodies are nothing but like the storage unit where they reserve the material in prokaryotic cells in the cytoplasm in the form of inclusion bodies. So in the cell, whatever the material is there, they are stored in the form of inclusion bodies. All right. They do not have any membrane bound to it. They are only lying in the cytoplasm. And examples of them are phosphate granules and synophysin granules and gas vacuoles. All right. So from this page, what did we learn? In prokaryotes, ribosomes are associated with the plasma membrane of the cell. Their size is 15 nanometer to 20 nanometer, and they are of having two subunits, 50S and 30S. Together, they form 70S in prokaryotic cell, and then they are the main site for protein synthesis. There are a lot of ribosomes together, and then what happens? They attach to a single mRNA. They attach to the single mRNA, and then these ribosomes are called as polyribosomes or polysomes. And these ribosomes, what do? Is they translate the mRNA into proteins, and that is called as translation. We can see inclusion bodies in prokaryotes where they do not have a membrane bound to them. They are found lying freely in the cytoplasm. Okay, and their main function is to reserve the material. Their main function is to reserve the material. All right, example is gas vacuoles, phosphate granules, and synophagal granules. Now let us talk about eukaryotic cells. All right, so eukaryotic cells include protists, plants, animals, and fungi. All right, and there is extensive compartmentalization of cytoplasm. What does that line mean from your NCRT is there are a lot of compartments, means there are a lot of cell organelles in the cytoplasm. All right, and they are membrane bound organelles. Okay, they are membrane bound organelles. Now, the first organelle that we are going to study is of cell membrane. All right, so we are going to study a little bit about cell membrane. So, let me tell you something. A scientist names are very important according to your NEET perspective. So, remember this scientist name. It is Singer and Nicholson. Singer and Nicholson came up with the fluid mosaic model in 1972. All right, Singer and Nicholson came up with this cell membrane model called as fluid mosaic model in 1972. Okay, so now I'll be explaining you guys about fluid mosaic model. First, I'll be explaining you with the help of the notes, and then I'll be showing you an animation so that you'll understand it more better. Okay, now what is the main function of cell membrane? And first of all, let me tell you something. Every cell has cell membrane separating it from the outer environment. Every cell has a cell membrane separating it from the outer environment. So now you have, how does this cell membrane look like? Is it actually, it has a biphospholipid layer. It means it looks something like this, like how I'm drawing on the screen. So it is biphospholipid layer. All right. Now, the main functions of the cell membrane is to be a barrier keeping the constituents of the cell in and making sure that unwanted substances do not get in. So we can say that cell membrane is semi-permeable in nature. Now, what does that mean? It means it only allows few things to pass through and few things it does not allow. All right. So what does it exactly do is, suppose think this is a cell and this is the cell membrane. Okay. Now, if there is a deadly substance that wants to enter, then it does not allow that to come inside. But if it is water, oxygen, vitamins, all the things that are needed, it will allow them to come inside. That's because it is semi-permeable. All right. So basically, it is a gate allowing transport. It acts as a gate allowing transport of essential nutrients and movement of cells, and even it allows to get rid of the waste products. So we can say that it acts as a gate, that it helps in transport of essential nutrients. I'm sorry about the background noise. Actually, there is construction going on, but I hope you're able to hear my voice. In case you do not, please let me know so that I will make a video again, like a mini review kind of a thing. So right now, what did we learn till here? Who proposed this fluid mosaic model? Who proposed fluid mosaic model is a neat question. All right. So Singer and Nicholson in 1972 proposed this model. Okay. And it is biphospholipid layer. All right. And every cell has a cell membrane to protect itself from outer environment. The functions are it is semi-permeable and it acts like a gate. That is, it allows oxygen, like water, vitamins, essential things to come out, and it also helps in getting rid of the waste materials. All right. So cell membrane is mainly composed of, we have already talked about it, biphospholipid layer means cell membrane is composed of lipids. All right. And lipids are arranged in such a way that this is the head, which is hydrophilic. Hydrophilic means it attracts water. This is hydrophobic tail. Okay. Let me tell you something about humans. How the cell membrane is there in red blood cells, that is erythrocytes. So erythrocyte cell membrane has 52% of protein and 40% of lipids. All right. It has 52% of proteins and 40% of lipids. Now, so it also has other kind of molecules like carbohydrates, it has proteins, it has cholesterols also. Okay. Now, if these two cells has to communicate, who helps in it? The cell membrane helps in communication. Okay. So suppose if this cell wants to give some of its constituent to this cells, so what does it do? It gives it out through the cell membrane and this cell accepts it. Okay, this process is called as endocytosis. And like if the sperm has to fuse with the egg, even that is possible because of the cell membrane, because it helps in fusion of proteins. So I'll be showing you in the video and animation. There are specific channels like, sodium potassium channel will only help in transport of specific materials. All right. Then you also have different channels. You have channels for peripheral proteins. You have channels for peripheral proteins, and then you have glycoproteins, which are there over here. Okay, then you might have cholesterol over here. So basically, it is a mosaic pattern. Like there are different kinds of things that are present inside the cell membrane. Okay, most complex components. So which one is the most complex components? Most complex components are proteins, and one of their main job is to make sure that right molecule comes in and right molecules goes out of the cell. Now, which are the molecules that can easily cross this bilayer is non-polar molecules, that is oxygen, carbon dioxide, and vitamins, which are non-polar molecules, can come inside easily, whereas polar molecules or charged molecules cannot cross the cell membrane. All right. So now let me show you the video so that you can easily understand.